Hello and welcome to World Science Festival Brisbane 2022. It's a wonderful world, all about biodiversity and living interconnectedness. I'm Bernie Hobbs. I'm rapt to be here with you.
Oh, I'm Rob Bell. I'm also rapt to be here with you.
He's doubly rapt. He's super rapt. We're all so rapt. We could only be rapter if there wasn't a stupid flood keeping us apart. We were so excited about seeing you all live in person. But look, this is going to be brilliant.
The next best thing?
Yep, totally. And not only that, we have got 3 gun scientists who are not going to tell us and show us, but have a lot of fun getting us all jazzed up about biodiversity and the interconnectedness of the living world. Are you feeling jazzed up already, Rob?
Yeah, definitely. I'm jazzed up, I'm rapt, I'm excited, I'm
You've got every musical metaphor going for you.
Yeah, that's true, yeah. I'm not going to rap, though, because that will end badly.
No, that's never a good thing for a dad. No, never. Okay, so before we get started, I do just want to do an acknowledgement of country. So in keeping with the spirit of reconciliation and on behalf of Queensland Museum Network, we acknowledge the First Peoples, the traditional owners of the lands where we live and work. We recognise that their continuing connection to land, water, sky and community. And we pay respect to elders past and present and acknowledge the important role Aboriginal peoples and Torres Strait Islander peoples continue to play within World Science Festival Brisbane and the Queensland community. And you couldn't get a better summing up of biodiversity and the importance of it than you get in the connection to country that we all share as well. So, Rob, I've been just jittering and being so excited about this all morning. So let's just not waste any time. As I said, we've got 3 gun scientists, and I'm going to head straight over now and meet our first one. Associate Professor Guy Kessler. We'll be back to you in a moment, Rob.
Brilliant.
All right, here we go. Off to meet Associate Professor Guy Kessler. Now, Guy, you have got an incredible background because you started, everyone who loves animals in Australia, we think kangaroos, koalas, that sort of thing. But you grew up in South Africa and your first animals were not kangaroos or koalas.
No, they weren't. I did a fair bit of work in national parks and I would be working on things like elephants and rhinos. There were a few close calls. I had to run away from a few rhino once or twice, but yeah, quite a different kind of dynamic.
All right. But you're going to be talking to us. I mean, you've been in Queensland for a long time. You've been at Griffith since 2005. Yeah. So, you've learned how to pick a koala from an elephant at this stage.
They're fuzzy.
Yeah. You're a bit of a legend at it. But today you're going to talk to us about how scientists study and understand these interconnected groups of organisms living together, ecosystems.
Yeah, biodiversity in general, So just a quick overview of what it is and why it's important.
And jelly beans
and some demonstrations.
And beakers and funnels. What's not to love?
Thanks, Bernie. Yeah. So, essentially, when we start thinking about biodiversity, we can kick off with our world, our global world. we basically living on Earth 1.0. There is no planet B. We need to conserve the environments and the species. And our planet is not only natural biodiversity that we're looking at, we're also thinking about cultural diversity, so thinking about our traditional owners, for example. So our planet, biodiversity is the connectedness of all that. So when we think about it, we need to find out what it is, why it matters, because it's really important, and then how people can actually get involved in learning more about biodiversity and contributing to biodiversity knowledge so that scientists like us, for example, can actually do something with it.
And you're going to give us some hints on how everyone at home can
how we can actually get involved and get your hands wet and dirty, for example. So I'm going to run through what all these different facets are. So let's start off with, what biodiversity is. Okay. So back in 1990, when we start thinking about what biodiversity is, Reid Noss came up with this framework about the different components of biodiversity. So we have compositional, functional, and structural components. So if we go to the next slide, we have all these different elements. So in that diagram on the left-hand side, we can see all these core areas. We've got the structural, functional, and compositional. Now I've simplified it a little bit and put these big black arrows in for 2 of those. So the structural one, for example, is if we start thinking about, say, the structure of a forest, where we start and it's you have shrubs and grasses at the small end, and then you have understory vegetation to tall canopy trees, etc. So that's kind of structure. When you think about function, it's about the services that the plants and animals might provide. So the functional diversity, if, for example, you were looking at a bird, that bird might disperse a seed, so that's a function that it performs. You might have a bee that pollinates a flower, it's the same. It's a functional kind of role that it plays within the community.
And the plants provide those seeds.
And the plants provide oxygen, etc. So there's functions and structures that they provide. I'm going to focus more on today's exercise around the compositional components, and that's why I've added in a few extra words into the slide over there. So
in 0.4 size font, but no. That's...
So at the smallest level, so all of those little circles that you see in that diagram represent the different scales of biodiversity. So we can start off really small, and then we kind of get bigger and bigger as we start looking at bigger and bigger things. So if we think about compositional biodiversity, what we're really looking at here is, at the smallest level, at genes. So we're looking at the DNA that makes us up. So each of us has DNA inside us. It tells us, what colour our hair is, what colour our eyes are, how we behave, etc., etc. All animals have those genes. And essentially, if we step up from that genetic component, that's our genetic biodiversity, we then start looking at individual species. So the spider image, for example, that's a species that we have that we're looking at. And if we have a certain number of individuals of that particular species, we refer to that as a population. So if we have 10 individuals of a species, that's a population living in an area.
I hope there are no arachnophobes watching, because a population of spiders...
That's such a cool spider, though. Those trapdoor spiders, just really funky.
They're gorgeous.
Once we start adding more species to our area, so we have a range of unique species, different types of species in an area, then we start referring to that as a community. So each of those species has a different number of individuals that are within them, and they all live in different areas. They perform different functions, and they have different, or they live in different components of the habitat. So some of them might live on the ground, others might live in the canopy, etc. That then is referred to as our community, and they're living in an ecosystem. And then at the biggest scale, you can add a whole bunch of ecosystems together so that you're looking at a large landscape. And that essentially is how all of the different components are connected when we start thinking about biodiversity.
So they just get more and more complex as you add more.
So when we're thinking about this, because it's so complex, you're learning about biodiversity is really complex. We need to try and make it a bit more simpler. So we start using what we refer to as biodiversity surrogates. So now a biodiversity surrogate is something that we can use as scientists that represents the diversity of life in a particular area. So we can go and have a look at, in this example, I've got birds up there, so we've got rainbow lorikeets and yellow robins and top-knot pigeons, etc., and scarlet honeyeaters. So they represent a component of biodiversity, birds in this case, and their species. So often scientists use species as surrogates for biodiversity because they're easy to find. We can go out and we can say that's a species, that's a species, that's a species, and we can find out what it is. Takes a lot more effort to go and find out what the genetic diversity is. We can do it, but it's a lot more effort. So we use these biodiversity surrogates to go and find out. So let's find out about what the different species components are. So there's some key elements to a species. For most of our higher level taxa, so a taxon is a scientific classification system. So we start really low at low taxonomic groups, and we end up with the higher taxonomic groups, which are vertebrates, for example. So all the things that we can actually learn most about. So we know a lot of information about our higher level taxonomic groups. Things like mammals, birds, reptiles, frogs, plants, some of our invertebrates, but not so much about fungi, bacteria, all those other kind of smaller things that people don't know a lot about. But for each of these species, we can actually go and identify 3 important components.
I knew you'd get an elephant in there, yeah.
Absolutely. So that's in the Kruger National Park, actually. So you can see there with those photos, there's a whole range of diversity. So, there's invertebrates, there's fungi, there's plants, there's invasive species, there's everything. So all of those species, each unique species, if we count up all of those, we have what we refer to as the richness of our community. So the species richness just relates to, well, how many different species do we find?
So how much diversity there is.
The next component is how many individuals of each of those species are there, and that relates to how abundant they are. So some species may be really abundant, so you'll have lots and lots of individuals. So cane toads, for example, might be really super abundant, and there's just hundreds of them wherever we go. Other species may be less abundant, or uncommon or rare. So the abundance is a really important component in knowing what we want to learn about biodiversity. The other component is, well, once you know where the species, which species you have and how many there are, you need to know where they are. So that relates to the distribution. So understanding where the species lives, what kind of habitats it needs, all really important in how to actually go about conserving them. So if we can understand which species we have, how the communities are made up, how they interact with each other, and where they live, we can then protect those environments going forward. So to do this, we need to do what we refer to as surveys. Mm-hmm. So we have to go out into the habitat and actually go and run some survey transects. So in this image here, for example, I'm doing an actual bird survey. I'm using binoculars. It's a visual survey. I'm walking through the forest.
I love that you've got a clipboard and pen too,
Absolutely.
in case any birds have really strong opinions.
Old school, you know, still pen and paper. I'm not even using tech there. So different types of animals will have different types of surveys. So for a bird, I'll be using binoculars and visual cues and auditory cues. So for some birds, I'll identify them by their calls. For other types of surveys, I might be doing something else. So you could be doing wildlife camera trapping for a large mammal. You might be setting pitfall traps for reptiles. You might be dip netting for invertebrates in a pond. You might be doing seine netting out in the beach if you're doing a marine scientist, for example.
Seine netting?
Seine netting.
Okay.
It's where you go out and basically pull a net through a kind of a tidal area and you catch some fish and other bugs and things.
Okay.
So the type of species you're looking for will dictate what kind of survey you're going to be doing. And the information that we get from these surveys allows us to make comparisons of the habitats that we're in. And so for today's exercise, what I'm going to be doing is a little habitat comparison like this. So in this illustration that we're going to use, we're going to compare a nice intact habitat, a nice rainforest stream environment that's relatively well protected. It's in a national park. And we're going to compare that to an urban pond, which maybe is impacted because there's a lot of rainwater, as we know recently with all the flooding. And there's pollutants and other contaminants, invasive species that might be living in there that impact on that aquatic environment. So we can compare these two habitat comparisons. And I'm going to do that today using jelly beans. So we all love jelly beans.
We all love jelly beans, and we want to protect their diversity.
The jelly beans essentially represent the species diversity. So each different colour in the jelly bean represents a different species. And for our example, our jelly beans represent frogs because, you know, the number of frogs that we might find in a particular habitat is probably the same number of colours we have of jelly beans in a certain area. So that's if we wanted to do birds, I'd need to find 40 or 50 different coloured jelly beans. Anyway, so we're going to run this little exercise. So I've got two habitats here. This bucket here with these jelly beans is my intact forest habitat.
So that's like Daintree River up
in the
Daintree Forest?
Daintree or Lamington National Park.
Lamington National Park, good, Lamington.
Or the Daintree. Yep. This one could be an urban park, any urban pond somewhere or an urban creek.
Kedron Brook.
Yeah. I don't ever been there, but yeah. So this is our urban park that we're going to do. So what we're going to do is going to empty these into our trays here.
There's a lot of jelly beans. I think we might need that.
And we're going to sort them out, really. So
I reckon we need help. Dr. Rob, get on over here and put on your best jelly bean sorting.
Each colour represents a different species, so we need to separate out all the colours into the measuring cylinders in the front here. No, we're not going to eat these ones.
I know, stay with it, Rob. Stay.
Right,
you're going to get a sugar high if you eat these.
So look, there's one left in the pond.
Okay, you don't know what it's been in. So
we're going to separate out all the colours.
So you're sorting. Oops, I just got a white one in there.
Keep it separate. I can do that. Okay.
So
we don't want to mix up our habitat.
there might be a slight error in my sampling here, Guy.
Yep.
Yeah, I see that.
Rob's scientific error already. You've put in the wrong
cross-pollinating here.
Frog counter. Yep. I need to do a better job at this.
Yeah, that's okay.
What frogs would you typically find around?
In some of our rainforest habitats, we'll get things like great barred frogs. So they're quite big and noisy. They're quite common. So if you go for a spotlight walk, often frogging is a nocturnal activity because most of the frogs tend to be active at night.
And can you tell frogs like just by their song mostly, or do you have to actually spot them?
No, so during the breeding season, the frogs will all be calling, and you can actually identify the frogs from their calls, which makes it really easy. So some calls are a lot easier to recognise than others because the frog calls can be quite similar.
There's one that sounds like a motorbike. I heard it on radio the other day, the motorbike frog. Yeah.
So, they are. You can definitely
You don't just live near a bikey compound?
I do indeed, but I also heard this on radio. No, I'm getting close. How are you? Can I just say, 2 against 1,
You're
and I'm
actually winning, I
smashing
think.
it.
But I
Yeah.
feel like there are more frogs over here. I could be wrong. We'll
are
find
more
out once
frogs.
this survey is complete.
Yeah, okay. Oh, I'm down to, I need this one. Oh, okay.
Yeah, well, and let's put it this way. When you're doing a frog
All right,
survey,
there's a green one
the
stuck in
total
there.
abundance of frogs that you're going to be finding probably not as high as what we're getting here with the jelly beans.
Oh, no. That's all right.
all
Add those frogs to those frogs.
So who wins? Oh, I do. And the prize is?
You finish your survey first, but I think we're finding more frogs over here in our environment.
All right, well, I just rescued one black frog
Oh.
from
We're
this
almost
side of the
done.
table. Do you want a hand?
Species migration
Yeah.
going from
All
the
part of-
urban environment.
Oh,
So-
I'm already seeing a bit of a pattern.
Yeah, well, these ones that I'm collecting at the moment don't seem to appear in your habitat for starters.
No, that's right.
It's all right. Yeah, so then we have all of our frogs. So this is this habitat over here. This is our intact rainforest stream. And what we can tell already is that we have 6 species in this stream environment. This one is our most abundant one, followed by that one, and then this one, the green one, sorry, and then the blue one. So that represents, our most dominant species. So using abundance, we can actually find out which species are dominant or rare, for example.
Yeah.
So these ones are probably less common. These ones are dominant ones. But there's a fairly good representation across all of the different species that we find.
And that's pretty good,
So
surely.
this one might
6
be one
different
of our
ones.
great barred frogs. This one might be an eastern sedge frog, for example, because they're green anyway.
Yeah.
This one could be another tree frog, etc. So in our urban environment, you'll notice that we have fewer species. So we only have 4 species in our urban environment. We do notice that there's an overlap. So 2 of our species are the same that we get in our. So these could be relatively widespread or common species that we find that are found in multiple habitats all over the place.
But
So
they're
they
not
could be-
doing well
No, they're not doing
in
as
this
well as
one.
what they would be doing in a natural environment, for example. And then we see that there's some super common ones. This one,
Yeah, I think
for
I know what
example,
that guy
this
is.
one you might have a guess. This, for example, might be the cane toad.
Old cane toad.
Yeah, so
Yep.
one of our invasive species, and if you do go around in the places, you'll tend to find a lot of these guys around. So
But that might not be invasive. That might just be a
it could
native
be
one
a common frog. So that might be something like a striped marsh frog or something like
Right.
that. Maybe copes quite well with those urban environments. It's adapted
It's a
enough
rock and
to
roll
survive.
frog. Yeah, it just lives on the edge.
So the thing with that, so what we can do then is, this just illustrates how we can differentiate the different diversity components, looking at the number of species, the abundance. The next thing we need to do is look at how we actually go about surveying them properly in our environment. So there we do what we refer to as transect surveys.
Because you wouldn't just do this once and
No.
go, that's the answer. Yeah.
Once isn't enough. You need to go out multiple times to get a good representation of what you actually find in your environment. So if we look at how we do surveys, so I've got another demonstration that looks at how we look at that. So if we're surveying our communities, so in this example on the screen, I've got a gold box, and in that box, each of those coloured circles represents one of our species, and the black line or the arrow through the box represents a transect that I'm doing. Now, a transect could be anything. So for frogs, it might be walking along a creek line, and we're recording how many frogs we see or hear along that creek line. For birds, it would be a transect through the forest. For kangaroos, it might be flying in an aircraft for 20 kilometres and counting how many kangaroos you see on each side of the aircraft, for example.
So transect just means it's one
It's a line.
measurement go. Yeah.
So typically when you're doing surveys, you have multiple transects, so we call that replication. So you want to make sure that when you're doing your surveys, you put enough effort in to find the species that you're wanting to find. So we call that replication, and a good scientific design needs a lot of replication. So
Because you might miss something. Yeah.
so you need to replicate how many unique sites you're surveying. So you can't just go and survey one stream. You need to survey 10 or 20 streams. You can't just have one site in that stream. You need to have multiple sites in that stream. Okay, so if we run a second transect, so you'll see that as we add more transects in, the number of species that we detect and the numbers of each of those different species, represented by the colours, start changing. So if we add a third transect in, our species richness will go up to 8, for example, and the pie graph on the right, the numbers inside that chart illustrate how many more coloured circles are being hit by that line. So those essentially are frogs that we find along the way. As soon as the line hits that particular dot, we record that as a frog that we've seen.
So it's getting more accurate in picking up more species and also showing the proportions of the different species.
And that then represents this kind of representation of the relative abundance of each of them and which ones might be dominant or rare, for example. So if we add in one last transect, we can see that our total number of species adds up to 9, and the red ones come out as being the dominant ones, the black ones come out as being the next one, then pink, etc. So they obviously don't represent these colours here.
And that was just one go. We did all right.
Yeah, not too bad. But what you might have noticed is that, so I know that in that particular community that there is a really threatened species, and threatened species are those that are really rare in the environment, they have specialised requirements, and I don't know if you can pick it out, but there's a circle in that between rows transects 1 and 2, in the middle of the screen there's a circle there that looks gold. That is a rare and threatened species. So that would represent, there's only one individual in that entire community, and we didn't pick him up. Okay, so this just is an illustration, I guess, that we need a lot of effort to detect our rare species. And so just 1 or 2 surveys isn't sufficient to go and say, well, can't find that species there. So just how we go about doing it. So threatened species, really important. We may know a bit about them. So coming to Australia, one of our key iconic threatened species is the koala, and that species is listed as threatened nationally. We could have threatened ecological communities, so actual vegetation communities that are threatened entirely, and then some other less well-known threatened species like the glossy black cockatoo. So that's the bird that's on the right, and that's actually a species that I do a bit of my research on. So that's me setting up some acoustic recorders in the landscape to go and find that particular species. So collecting information is really important. So this is where you can get involved. So how do we get all that information? Many of us might be an outdoor naturalist and you want to get involved in citizen science. So there's a whole raft of mobile apps that we can actually use to collect this information. In Australia, we have some dedicated ones. So Bird Data is an app that's run by BirdLife Australia, and that's obviously to collect information about birds, where you see them, which species they are. We have the Frog ID app, that one's that collects information about frogs. And then there's international apps like eBird and iNaturalist that allow you to collect information about birds, but otherwise iNaturalist is one that you can collect information about everything. And the iNaturalist app is connected to this new Seek app that actually allows you to use your smartphone. So you can go out, find an animal, take a photo of it, and it will try and identify it for you based on the actual photo. So if you don't necessarily need to know a lot about what you're finding, because the apps these days can try and identify it for you.
And it works for plants as well, I think, because we've used it in our regen group. So
let's show you a bit about some of these apps. So one of the ones that we can use, the Frog ID app.
I love this.
This is an app that was set up by the Australian Museum. And the way it works is that it looks to basically have you going out and listening to frog calls. So the frog calls, essentially, this is a green tree frog. It's a photograph of it actually starting to call. So it'll start bulging out its air sac here and start calling. And we can use those calls to then get information. So the app, what you do with the app is that's a little rubella, Latoria rubella on the left-hand side. It's busy calling. You record the app, so that's a screenshot of the app actually recording the call. So you record it for a certain amount of time. You make some notes about the habitat that you saw the animal in. It's whether it's a natural pond, a creek, etc. And then it gives you some options about which species it is. You listen to some of those and you say, I think it's this one or this one, and you submit your record to the scientists and they go, yes, it's this particular frog, and they add it to their list. So that information, once they use all that information, you can generate maps of where you find all the frogs. So this is Brisbane with all the verified frogs that we've seen.
Wow.
Each of those green dots represents a frog identification. And if we zoom into South Bank, which is where we would have been located, that shows you exactly where the frogs have been recorded, which frogs they were, when they were recorded. So we can get a lot of information from those records. We can find out which species it is, how many individuals there were, how frequently it was calling, when it was calling, was it during the day, what time of the year, etc.
And then by comparing that year upon year and across the area, you can work out what's happening with the population and how healthy the biodiversity is.
So the iNaturalist app is another one that we can use. So that differs because on the right-hand side here, you'll see that that's again a snapshot of the iNaturalist records for Brisbane, and you can see there's millions of them, or hundreds of thousands of them. It's
Don't go billions, yeah.
213,000 records from 4,000 people. So
Wow.
the engagement with citizen science is huge.
And the great thing with citizen science, you just have to be a citizen, like you just have to be a person, and don't freak out if you're not sure if it's right, because scientists are checking it and making sure that you get it.
So yeah, the engagement's huge, and you can record anything. So there's birds, there's plants, there's anything being recorded on that particular app. And then the question that people might be asking though is, well, it's fine, submitting a record to some of these apps and saying, well, but what happens with the information? So this is where we can actually use that information that citizen scientists generate and start learning more about species, like glossy black cockatoos, for example. So this map gives an illustration of how we use citizen science records to map where good glossy black cockatoo habitat would be. So we used all the records, and all those green areas on the map represent areas that are really good high potential habitat. All the pink areas are possible areas that we can see them in. But that was all based on citizen science records.
You would love that map because it means you get to go to Straddie all the time to study the bird.
Our project that we're working on is taking us up to Straddie pretty soon, hopefully. So yeah.
Nice.
Essentially that's a great example about how people can get involved, and you never know what you're going to find.
Yeah.
So, when you're out there at night looking for things, there could be any type of creature that you could find. So get involved.
Even when you're not already a scientist, you can be a scientist by being a citizen scientist and help real life
scientists. And you might
scientists.
find something like this. If you go out with a UV torch, for example, different types of surveys, you might find that things fluoresce.
What is that?
It's a millipede.
Wow. Do all millipedes fluoresce?
I wasn't aware of it, but it looks like they do. So that one certainly does.
Wow. Okay, disco maggot.
Millipedes,
it's
scorpions, under UV light. Yeah, amazing. So you get involved, get out there.
Brilliant.
Start recording species.
I love it. Yay, thanks. Associate Professor Guy, that's brilliant. We're so glad you came to Australia to help us out. Dr. Rob, what have you got?
Thank you very much, Bernie. Kimberly is going to be telling us a whole lot about biodiversity as well, but we're going to be jumping sort of from the freshwater ponds to somewhere completely different. What sort of creatures and biodiversity do you research, Kimberly?
Well, I research sea turtles. They're one of my favourite animals.
Perhaps why we have a turtle shell behind us. We can find out a bit more about that. I guess, what specifically, when you're researching sea turtles, do you look at?
So I research the effects of contaminants on sea turtles, but I've been working with sea turtles generally for about 7 years.
All right, well, why don't you tell us all of the cool general stuff about sea turtles, and I'll throw in a few questions along the way. How does that sound?
Sure, sounds good. So sea turtles are marine reptiles, and there are 4 types of marine reptiles. There are snakes, lizards, crocodiles, and sea turtles. And these guys are all specially adapted to life in salt water, so these guys are pretty unique. And sea turtles are ancient. They're over 100 million years old, which means that they share this earth with the dinosaurs.
Cool.
And some of these ancient species were absolutely massive. Like take Archelon here, who was over 4 meters in length. So that's more than double the average human height. And it's estimated
That's crazy. So the other, is there a modern day turtle? Is that the far
Yeah, the one at the far right,
so,
the leatherback turtle.
Whoa, Archeon. Yep, don't want to see one of those out there. That would be pretty scary.
So it's estimated that there used to be 30 different types of species, but today we only have 7 of them. So we've got our leatherback, which is our largest at about 2 meters in length.
Okay. And that's about the same as me.
Yeah, exactly. And then we've got the hawksbill, the green, the olive ridley, the loggerhead, the flatback, and the Kemp's ridley. And we're really lucky here in Australia that we have 6 of these species, which includes the flatback, which only nests here.
Lucky us.
So some of these species are really easy to tell apart, like the leatherback and the green turtle. So the leatherback has these big ridges down its back, and the green turtle has these hard scales that form a shell. But some of them are harder to tell apart, like this loggerhead and hawksbill hatchling. So we use these special identification keys that help us identify what species we're working with. And it's largely based on the number of scales on their shell or carapace, as well as on their head or face.
The leatherback, despite its name, it still has a fairly hard shell or carapace?
Its skin is quite leathery, so it's pretty unique amongst the turtle world.
Yeah. Very strange. Okay. Sorry.
So sea turtles have a very complicated life cycle. For one, they move hundreds or thousands of kilometers throughout their life. And 2, they are very long-lived. So this is the size of a sea turtle when it comes out of the nest.
Wow, that's so Yep, that's a tiny, tiny little thing there.
Tiny little thing, yeah. And this is an example of how big they can get, or possibly even bigger.
Now, what sort of turtle was this?
This is a hawksbill turtle.
Okay, wow, huge.
Well, they get much bigger than this,
Yeah,
too.
okay, so this is a medium-sized hawksbill?
It's pretty big for a hawksbill,
Okay, for
but
hawksbill.
depending on
But
species, they
yes,
can
they'll
get a lot bigger. Yeah.
Yep.
So you can just imagine how long it takes them to grow that large. So one of the oldest turtles that I know of is about 70 years old, but we don't actually know how long they can live because they move around so much throughout their life and they live for so long. It's really difficult for us to study them through their whole life cycle.
Of course, yeah.
But we're going to go through the life cycle right now, one step at a time, starting with the nesting beach. So after about 6 to 8 weeks of incubation, the hatchlings start to emerge. And remember, they are so small, and they've got about 60 centimeters of sand to dig out of. And I think we've got a video of that here.
Oh, yes, we do.
Yeah, there we go.
Well, that is super cute.
So with so much sand to dig out of,
Yeah.
how do they make it out? Well, their nest is shaped like a giant funnel, and they all dig together, working together to dig their way out of the sand, which is why we see them all emerging at once.
Is there a particular cue for them? Is it just length of time? I mean, obviously they all seem to have hatched at the same time and dug up at the same time. Is it a time of day or?
Yeah, they actually wait for the sand temperatures to cool, so typically they emerge at night.
Yep.
And there's no parental care, so after they emerge, they run down the beach to the water to begin the next stage of their life cycle in the open ocean, otherwise known as the lost years.
Oh, the lost years. So like the teenagers.
Yeah.
Okay, got it.
So hatchlings enter what we call a swimming frenzy, where they swim non-stop for several days until they reach ocean currents. And then they float around in these ocean currents for about 5 years, and this is what we call the lost years because we don't know a whole lot about where they go or what they're doing during this time.
And that's just because they're really hard to track through that because they're tiny little things. It'd be very hard to sort of tag properly or have radios on or anything like that.
Yeah, exactly. And so after about 5 years, they'll come back and they'll pick a feeding ground. And these areas are typically close to shore, where there's lots of seagrass or coral reefs like the video we have here. And this is going to be their home. This is where they live for the rest of their lives. And they don't really consider leaving until they're about the age of 30 when they'll go on their first breeding migration, which is the next stage of the life cycle. So males and females will travel hundreds of kilometers sometimes to special breeding areas where they all aggregate together. And then they mate with several partners so that they can make sure to pass on the best genes to the next generation. Now from here, males get to go back to their feeding ground to rest and relax, but females will have more work to do. So they go back to their nesting beach. And this is actually the same beach where they were hatched that they go back to lay their eggs.
So this is before the lost years. So when they came out as those tiny little creatures, got lost. Well, they didn't get lost, but they swam around for their lost years. They're now, what, we're talking 20, 30 years later.
Yeah.
They come back to the same beach. How on earth do they manage to do that?
Well, they use the magnetic fields of the Earth to navigate. So we're still not really sure exactly how they do it, but it's pretty fantastic. They come within, several kilometers of where they hatch sometimes.
And obviously they wouldn't all come back at once because a lot of them hatch, so I guess they wouldn't all come back at the same time. How many make it through, I guess? That's one good question.
We don't exactly know the answer to that, but it's about 1 in 1,000, we estimate. Yeah, so the female will come up the beach and she digs a hole that's about 60 centimeters deep, and then they lay on average about 100 eggs. And then she'll spend a lot of time covering up the nest and camouflaging it, throwing lots of sand. Trust me, I've gotten a few facefuls of sand. And this because, remember I said they don't have any parental care, so she really wants to protect the nest from predators. And then she'll head back to the water. And she'll do this between 2 and 8 times a nesting season at about every 2-week intervals.
Wow.
So this is exhausting for females.
And the whole time the male is doing?
Oh, he's just chilling. Back in the feeding grounds.
It's good to be a male turtle, apparently. Okay, so She's doing all the hard work, which is great.
Yeah. So after she's done, she'll head back to the feeding ground. Well, she'll take a few years, maybe 2, 3, or 4 years to rest and recover before she considers doing another breeding.
So they don't breed every year, it's every however many.
Yeah.
Okay, interesting.
A couple of years.
Yeah.
So to put all this life cycle in an Australian perspective, so hatchlings hatched here in the east coast of Australia will swim out past the coast of New Zealand until they enter ocean currents where they'll float around for about 5 years. And then they'll come back to a feeding ground here until they're about 30 years old before they go on their first breeding migration. And remember, these can be hundreds of kilometers. So sea turtles feeding here in Moreton Bay can go all the way up to the Great Barrier Reef to nest and then come all the way back to Moreton Bay to live.
It's such a fascinating life cycle. Like it's mind-blowing.
Well, and because they use so many different habitats, they use the beach, they use coastal areas, they use the open ocean, and then everywhere in between, they're actually exposed to a lot of threats. Yeah. So some of these threats are natural, like predation and disease, but an increasing number of these are human. So habitat loss and degradation is a big threat to sea turtle habitats like the nesting beaches and the coastal areas they use. So it's really important for us to identify these areas where turtles nest and where they live so we can better protect these habitats from things like coastal development. The next threat is chemical pollution.
So this is sort of what you're studying, yeah?
Yeah, exactly. This is my area of research. So chemicals enter the environment through industrial activity, agricultural activity, and urban runoff. And they can be taken up by turtles through their food, where they can cause health effects. But we don't exactly know what effects chemicals have in our turtles, and that's the work we do at Griffith. We're trying to answer those questions. So the next threat is marine debris. Now, I'm sure we've all heard a lot about this lately with plastic items like plastic bags being ingested by sea turtles. And you can see these plastic bags look a lot like jellyfish.
And they like to eat jellyfish, presumably.
Yes, they do. And sea turtles can also become entangled in these things like fishing nets or fishing line that have been discarded. So it's really important that we avoid single-use plastic items to prevent this kind of marine debris.
Well, particularly when there's only, say, 1 in 1,000 making it to adulthood.
Yeah.
We want them to last as long as they can as adults, right?
Exactly.
Yeah, absolutely.
We really want to help them out. So the next threat is light pollution, which is something we might not have considered.
No. So how is light a threat to a turtle?
Well, let me give an example of light pollution first. So if you're in Brisbane and you look up in the night sky, you might only see a handful of stars.
Sure.
But if you go out bush, you're going to see a lot more, and you might even see the Milky Way. Well, that's an example of light pollution. So on a nesting beach, artificial lighting can actually deter females from coming up to nest at all. And when hatchlings emerge, they're naturally drawn to the lowest light horizon, which is over the ocean. But on a beach with a lot of artificial lighting, they can actually head inland, never making it to the ocean.
So if there's a car park with like street lights or something, they might head up towards that.
Yeah, exactly. So in areas where turtles nest close to where humans live, like in Bundaberg, the Cut the Glow campaign is really helping to raise awareness about this issue and reduce light pollution. So the next threat we have for turtles is human consumption. Yeah. Now, I mentioned natural predation, but obviously they're predated by humans as well. So sea turtles are hunted for their meat, and also their eggs are collected for consumption all around the world. In many places, this is illegal, but it still happens as a source of income for humans. And in these places, we might consider using ecotourism surrounding the turtles as an alternate source of income instead of a legal take. But in some places, it's culturally significant for them to consume turtles and their eggs. And in these places, researchers, governments, and locals can work together to make sure that this harvest is sustainable for generations to come. And sea turtles, especially hawksbill turtles, like this guy here, are harvested for their carapace, which is used to make tortoiseshell items like jewelry.
What's the carapace made from? It feels like bone, but...
It's actually keratin, so the same things our fingernails and our hair.
Wow.
So they use it to make jewelry, which Yeah, it's really
I've seen a lot of
beautiful.
fake tortoise shell stuff as well, so I guess telling the two apart might be difficult.
Yeah, exactly. And there's lots of tricks that we can learn so we can do this, because a lot of people might not even realize that they're
What they're
buying
getting.
a turtle item.
Yeah.
And the last threat to sea turtles is that they're taken up. Oh, not the last threat. So turtles can be hit by boats in shallow areas where they live, and people like to drive their boats. And this can cause the carapace to crack or be cut, which can often be fatal. So it's important for us to know where exactly these collisions are occurring so that we can implement things like go-slow zones to reduce the number of fatal collisions. And so the next threat, our last threat, is incidental capture in fishing gear. So thousands of turtles are taken every year this way.
So this is sort of commercial fishing gear, right?
Yeah, exactly. Yeah, but it's not all doom and gloom. One of the best conservation success stories we have for sea turtles actually comes from fisheries and the introduction of the turtle excluder device. So in northern Australia, the northern prawn fisheries overlap with turtle habitat. And in the 1990s, this resulted in 5,000 to 6,000 turtles taken a year.
Wow.
And when they're captured in trawl nets, they can't make their way to the surface to breathe, and they end up drowning. So then comes the introduction of the turtle exclusion device. So this is basically a big grill that they set in the net in between the trawl end and the catch end that allows small things like prawns to go all the way through, and big things like sea turtles are prevented from passing through and instead are released through a small opening. But I actually have a turtle excluder device here.
Excellent. I was going to say the diagram is good, but let's do a little demonstration.
Yeah,
this is it over here. This looks like commercial fishing gear. So we have a turtle exclusion device.
And we have a turtle volunteer to help us.
Excellent. I thought we needed a turtle. Which way around? There we go.
Yeah.
You grab that end. Oh, hang on. Let me go and chat to our turtle, if I may, just quickly.
Yeah.
I'll give you a hand.
Thank you, Bernie. Excellent. Hello. What's your name?
Piper.
Hello, Piper. And have you been in training long for being a turtle? Think it's a pretty easy job?
Yeah.
Well, I guess you'll find out shortly. So tell us, Kimberly, what we're looking at here. So this is the end that is being dragged through the ocean, is that correct?
This is the trawl end that's being dragged from the boat, and that's the end that's catching all the prawns.
So Bernie's where all the catches. So all the prawns and turtle are out here.
So if we come along.
Piper, turtle, would you like to make your way into the catch net and see if you can get out?
We'll catch our turtle here. She hits the turtle exclusion device.
Oh, that's the big grill, so hits that.
And then she's released through the opening at the bottom, and she can swim to safety.
To freedom.
Swim away to freedom, hooray!
So these devices are really simple and really effective, but fisheries were reluctant to implement them at first because they were worried about reduction in prawn catch.
Yeah, it's a commercial industry, I suppose. Yep, sure.
So researchers got involved and found that actually loss to prawn catch was minimal, but interestingly, they found that the quality of the catch was improved.
Oh.
Yeah.
Well, the turtle's eating them in there. Well, how does the quality get improved?
So they were prevented from going into the catch end and squishing all the prawns.
Just messing it all up, basically.
Yeah, exactly.
Huh.
Yeah, so once that benefit was demonstrated to industry, it was a lot easier to make these devices mandatory, and that's exactly what happened in 2000.
Look at the difference it's made.
Yeah, so these are our results here. So you can see our East Coast loggerhead turtle population was in a significant decline before the year 2000. With over 5,000 turtles taken a year, this obviously wasn't sustainable. And then in 2000, turtle excluder devices became mandatory with a 97% release rate. And immediately we see this population start to recover and continue to recover today. So this is just so rewarding for scientists and conservationists and turtle lovers alike to see that the implementation of such a simple device can have such a big impact on the turtles that we love. So what else can we do to help our turtles? Well, for one, don't litter. Don't throw your trash in the habitats where they live, and consider using reusable items like water bottles and tote bags instead of single-use plastic items. We can use biodegradable soaps and detergents at home to limit the number of chemicals that are entering turtle habitats. And if you visit or live near turtle nesting beaches, be aware of light pollution at night and turn your lights off. And if you're really passionate, get involved. So my friends at Ocean Connect here have a ton of programs for kids of all ages to get involved in conservation. And finally, consider a career in science, because then you could be doing the research that ultimately influences policy and management and helps save our turtles. And if that wasn't convincing enough, here's how we catch turtles.
I think that was convincing enough, but I'm sure you've got an even better argument coming.
Oh yeah, well here's how we catch turtles. It's called turtle rodeo, and it's the absolute best part of my job.
So this is so you can put tags on them to monitor them, that sort of thing? Check their health?
Absolutely, sample them, do whatever we need to do. So
I see the turtle.
we drive around until we find a turtle, and we bring it up next to the boat.
How are you going to get to the turtle?
That's the fun part.
I think it's
then
coming.
we jump on them. We grab them underwater and we bring them to the surface, back to the boat, where we can tag them, measure them, sample them before we release them again.
Wow, turtle rodeo. All right, if that is not reason enough to take up science and start studying turtles, I don't know what is, frankly. Kimberly, thank you very much. It's been fascinating. Bernie, back to you.
So that was so great, Rob, and I'm glad we didn't need Piper the turtle out here to demonstrate turtle rodeos. Yeah, good thing you had the video, Kimberly. But we are on to our final amazing scientist here this morning. So we've heard about frogs in fresh water. We've heard about sea turtles in seawater. We're sticking with the water theme, and Professor Fran Sheldon, I'm just going to call you Fran from now on. Fran is all about fresh water, but none of those things. You're about the wiggly, crawly, flyy, buggy stuff in water.
The small stuff, the really important stuff, Bernie, to be totally honest.
Yeah. The stuff that I used to find in the bottom of our pool when we were kids and didn't look after it.
The stuff that the frogs eat, the stuff that the turtles eat.
All important for biodiversity.
All important for biodiversity, but these are the little guys and these are in freshwater streams. But before we talk about the bugs, I was going to tell you about water, because water's dominated our life this week in Brisbane and in New South Wales. Guy talked about frogs that lived in water, and Kimberly talked about turtles that lived in water. Water is super weird, and I thought it was pretty useful that we understood what water was. So water is this weird sticky substance, and you know, it's all around us in the oceans and now in Brisbane, and things that live in water have got some specific challenges.
And it's weird because you say it's sticky, but like it just doesn't feel sticky to us.
Not to us, it doesn't. So we'll explore some things about how it feels to tiny little animals. I want to tell you about some fun facts of water, first of all. So, did you know that all the water on Earth actually came from outer space when the Earth was very young? None of it was made on Earth.
Water is alien?
It's alien. All the water that we have was generated early in the Earth's life. And water covers 80% of the Earth's surface. So that's a lot of bombarding of things onto the Earth to give us all of that water. Fun fact 1. Fun fact 2. Did you know that water is the only substance on Earth that exists as a solid and a liquid and a gas at the temperatures that we find on Earth?
No.
Can you think of another one? Challenge me.
Cordial? No.
No, that's got water in it.
Okay.
And don't say Coke either.
No, obviously wasn't going to say that next. No.
So mercury is a really weird substance. You might have heard of that, but it's only a liquid on Earth. It's not a solid and a gas. So if you really stretch your brains, go away, homework, think if you can find something else that's other than water.
Yeah.
Strange stuff. Fun fact 3.
And that's why it's necessary for life. But anyway, we'll get to that another time.
Fun fact 3, the solid form of water is actually lighter than the liquid form, which is why ice floats.
Yeah,
now
so
that's really cool, because if ice didn't float and your streams and the ocean froze from the bottom up, what would happen to all the turtles in the northern part of? Not very good. They'd get frozen. It's really a weird part of water. Fun fact 4, water molecules are sticky, as you said. And that's we're going to explore exactly what makes them sticky, but that means things can walk on water. We've actually got that water strider in the slide, walking on the water, but it also means that water can climb up the small capillaries in plants, and we call them particular things, but our plants that gets from the roots right up to the surface, and the stickiness of water is just really cool. Fun fact 5, 66% of you, Bernie, is actually water.
I just had a drink, so it's probably 67.
Probably 67 now, a big gluggy mess. So water, a lot of you is water. There's water everywhere, and often we don't even know about it. So what exactly is water? So do you know?
H2O.
H2O. What does that actually mean? Well, you've got a great big oxygen, that's our green part, and 2 hydrogens, that's our blue part. Now, the weird thing about water is that the oxygen is negatively charged and the hydrogens are positively charged. So we've got this magnetic thing happening with water. It's a big magnet, or a little magnet, very tiny magnet. So we've got a positive end, the hydrogens, and we've got a negative end, the oxygens. And when you put lots of little water together, those positives, hydrogens, are attracted to the negatives, oxygen, and you actually get them all clumping. And so then that leads to this weird phenomenon where the positives and the negatives are attracted, and they call these things called hydrogen bonds. Now that keeps all these molecules together. So the physics and the chemistry suggest that water should be a gas when it's on Earth, but because these hydrogen bonds, they hold them together and make it a liquid for a long period of time in the temperature world.
And that's what makes it so sticky.
That's what makes it sticky as well. So we've got these hydrogen bonds. So let's just step through how water changes from a solid to a gas and how the hydrogen bonds work. So when water is ice, if we have a look at what it's like as ice, all of our hydrogens are each oxygen has 2 hydrogens attached, and it forms this sort of lattice, quite a rigid structure. That's ice. Then as the temperature increases, those water molecules start jumping around, molecules get all excited, they move apart, but they're attracted by these hydrogen bonds. So they don't go anywhere. So that forms our liquid, and it stays liquid for a long period of time. As you're boiling your kettle, you're boiling it and putting a lot of energy, and those hydrogen bonds eventually break. And that's when water becomes a gas and we get steam going up into the atmosphere. So I was going to see if we could get some people to demonstrate.
I so want to do this.
Do you want to be a water molecule?
It's
and
my
maybe
ambition.
maybe back in school or at home, you guys can be water molecules
Can we
too.
use a turtle? Yeah.
Yeah, can we have the
Piper.
turtle?
We need a
So don't knock over
Piper
my
water
bugs
model.
or they won't be with their water. And we might just skip to the demonstration on the slides of a water molecule. Now to do this, you've got to pretend that your heads are oxygens.
My head is oxygen.
And you've got to pretend, oh, we've got a big one here.
Sorry.
Oversized
Big water
water.
molecule.
Yeah.
And we've got to pretend that our hands are hydrogens, okay?
Okay.
So each hand, we have to have 2 hands on each head. So I'll go there
Okay.
and we'll go... Piper.
I need a head.
Right, all right. So now we're ice. So we're standing very still because ice is really quite cold. Now, as we start to heat up, we're going to start to jump around, but we're still cold, so we're going to go in on each other and get really close. Now we're water, but we're really cold. And did you notice how we're more dense? That's why ice floats, because it's lighter.
I'm getting punched in the ear.
Kettles
sorry about that.
Now you can get out now.
Oh, now we can
We're
go to a
getting
gas.
hotter, out. So when we become water, we're not too far away though, you're not yet gas, come back.
Sorry.
All right, so now we're liquid water. We're not touching.
Oh, sorry.
And now the kettle's boiling and now you're
Hey! Freedom.
We're all off.
That's great.
Thanks everyone for being water molecules.
Never seen better water in my life. Yeah.
So now we've got a bit of an understanding of how water actually works. We're going to be looking at how some of the things that live in water actually breathe.
Some of the things. Oh yeah. No, that's all
I'm
right.
not there yet. So when you breathe, what are you breathing?
I breathe in air. I keep the oxygen, and I spit out the carbon dioxide.
So most of our animals do the same thing. And so we need to think about how oxygen's actually held within water, because it's not like air out here.
Yeah.
So each of our oxygen in water, we've got less than 1% of the oxygen in water as we have in the atmosphere. So it's really hard.
because it's 20% of
21% out here, less than 1% in the actual water. So if we have a look at how oxygen actually sits within the water molecules, it actually has to sit in the gaps between the water molecules. So you can't get a lot of it in. So that's why it's less than 1%.
Yeah.
So it's pretty hard. So what we're going to do is we're going to have a look at some of our animals, and we're going to see exactly, can I move them over here so Bernie can, we need to share them.
Okay.
We're going to see exactly how some of our animals breathe. So we use our lungs.
Yeah.
And we know fish use gills.
Yeah.
And some of our very small things that live in creeks use a bit of a combination of both, and some other pretty weird things as well. So let's have a look. What kind of animal do we have up first? I think we have some snails.
Oh, yeah.
And I actually got some snails. I went out onto the campus at uni yesterday with some
this
helpers.
is all just from a pond at work.
This is from Mimosa Creek, which actually flows through the Tui Forest, which is where Griffith Uni campus
beautiful.
is. So snails are really weird. There's 2 different kinds. So one kind uses lungs, and you can see in the slide over there, the one with the lungs, and they do some crazy things. They come up to the surface of the water, and they twist their body around, and they grab a whole bubble of water and take it down. It's like a diving bell, like a scuba gear.
Yeah.
So we've got some of those in here, and that's exactly what they do. And you might see them if you've got snails in your fish tank at home. You'll see them hang around on the surface of the water, grabbing that air bubble. Other kinds of snails actually have gills, so they actually don't come and grab air, they just get their oxygen out of the water. Let's have a look what we have next on the slide. I think we have a mayfly. Now, I didn't get any mayflies in the forest.
Can't always be lucky.
Sometimes they're there, but with all the floods, the mayflies just weren't. Were probably just distributed over a big distance. But they have gills.
Right. Oh, they're
So what they do
beautiful.
is, they have these feathery things all along their bodies, and that increases the surface area of their body, and they can get more oxygen out of the water that way.
Yeah.
It's like having the lungs on the outside of your body, have
but
them all
more
flopping
attractive
around.
than having the lungs on the outside of your body. Yeah.
So we don't have mayflies. I think we have the next slide. I think we have the trichopterans, and these are caddisflies. Now I'm going to challenge you. Can you see something that looks like on the slide in the tray?
I love a challenge. It kind of looks like a long prawn, and no, I cannot see anything.
Well, that's because they're
that.
hiding in the sticks.
I was wondering why the sticks were walking around the tray. I didn't want to say anything about,
My moving sticks.
Yeah.
So the sticks actually have these
I can
caddisflies.
see little antennae or something, legs. Yeah.
And the legs will come out. So if you actually look on the slide, you can see that they've got all these extensions off their body, all the way down their body, and that helps increase the surface area for the oxygen.
A stick moving in a pond, you've seen a caddisfly.
You've seen a caddisfly, and they start off in really tiny little sticks.
Yeah.
And then as they grow, they've got to go and find bigger and bigger sticks.
Hilarious.
Yeah. And then eventually, when they're big enough, they close the stick off and they pupate and metamorphose and form that caddisfly, which flies
beautiful.
off and hangs out in the trees around the stream.
Oh, lovely.
So they're pretty cool, caddisflies. And some of them are even cooler because they make their cases out of sand grains. And people have worked out you can trick caddisflies, you can give them flakes of gold and precious stones. And instead of sand grains, they will make cases out of gold and stones. Jewellery, they're jewellery making flies. And then when the caddisfly leaves, you can wear them in your ears. All right.
If you like
Another
little
challenge. If
golden
you like
stick
little gold
jewellery
stick
things.
jewellery things.
Great. Wow.
What's another one? A dragonfly. Now, these are the naughty ones in the stream.
Because?
Look at where they breathe. So we're looking at different kinds of lungs. Dragonflies
I'd be
have
calling that their bottom. Yeah.
the bottom. Dragonflies have their lungs in their bottom.
Yes.
So they actually pump water in and out of their bottom to get oxygen. So they're bits of farty
So you don't
bugs.
want to swim behind a dragonfly.
Don't swim behind a dragonfly.
Always
They can-
be upstream of the dragonfly.
They can also use that to escape, so they can suddenly take off by pushing water out of their bottom.
Fart rocket.
We've got a dragonfly here.
Yeah.
So he's very cute. They're much more beautiful as adults.
Oh, yeah. They're pretty quick.
Yeah. And they've
Are you trying to make him breathe so we can see it?
no. But he will put his bottom up. So, bottom breathing dragonflies. What have we got next? And we have these other ones which we couldn't find in the creek. Normally they're in the creek, but they're the fly larvae, midge larvae they're called.
Yeah.
And you might have seen them in creeks. They're red. And you'll go and you have a look in a creek and you see these red little worms.
How long, like
They're only
millimetres?
about a centimetre.
A centimetre long. No.
They're often called bloodworms. And you can get longer worms, up to 2 or 3 centimetres. People can use them for fishing.
Yeah,
as well.
right.
Now, the red, interesting. What colour is your blood?
Red? Are you saying I'm a bloodworm? No.
Don't know, but no, I don't think so. But they have the same stuff in their body fluid as we have in our blood, which is iron.
Iron.
Yeah, and it makes their body fluid red. They don't have hemoglobin like us, but they've got similar. And what that does is it gets more oxygen out of the water than would normally be by having this iron in their blood.
That's genius,
Yeah,
isn't
they're
it?
pretty cool.
You wonder why it's not more of them using it. Like,
Well, interestingly, I do. We did get a yabby, but he was a bit naughty and he was trying to eat the other bugs.
that is such a yabby thing to do.
He's in solitary confinement. The
Little
yabbies
learning.
have blue. They're blue.
Ah,
and they've
they
got
are.
a copper substance in their blood, and
Yeah.
that does the same trick.
Oh,
So
wow.
if he hadn't been so bad, we would have brought him along. All right, other things that we've got in here that I don't think I've got slides for are these little green guys. These are
Beautiful.
damselflies. So they're like a cousin to dragonflies.
And that's not green because it's absorbing oxygen
No,
or
I think they're just
photosynthesizing.
because they've probably been eating some algae.
Right.
And so they go a bit green. But they've got gills at the back, so they don't
Oh.
bottom breathe.
Yeah.
They have these big plate-like gills at the back,
It's
which are
kind
pretty
of
cool.
bottom breathing.
Sort of
with
bottom
gills,
breathing.
Yeah.
And then we've got some cruisy little beetles flying
They're
around
very
in there.
fast.
They're cruisy. They don't have gills or anything. They just breathe through their body wall, which is quite
Oh,
interesting.
wow. So
And
they
there's-
don't do a bubble thing?
Some of them do, but these little tiny ones aren't
Yeah.
going to.
And sorry, do the caddisflies, do they use a bubble? Or?
No, they have the gills.
The gills,
all
that's
the
right,
way
okay.
along the body.
Yep.
These guys use a bubble. So
Right.
we didn't get any nodonectids or back swimmers. Sometimes you get them in your swimming pool, but they'll come up to the surface. There's a picture there of one sitting on the surface, and they grab a bubble of water. They have a hairy chest,
Yeah.
very delightful, and they hold the bubble of water against their hairy chest, and then they can breathe from that bubble.
Wow.
So they're not really breathing from the water, they're breathing from the atmosphere.
Wow, smart move.
Yeah, and if we think back to what Guy was talking about degraded habitats, Where we have really degraded habitats, we find more animals that get their oxygen from the atmosphere, like our nodonectids and some other weird ones that have a snorkel from their bottom. Here we have needlebugs, the snorkel bugs.
Where's
so
the
you can see the
snorkel?
snorkel at their bottom.
Okay.
Yeah, and they stick that up into the atmosphere and they get water from the,
Like a
from the
straw.
air, like a straw. So we find these sorts of things more in places where oxygen's likely to be a problem because of pollution or lots of organic matter or something. So bugs are weird, waters are weird.
Look, I have to say I never knew how weird until I knew what they got up to at the nether regions of themselves.
That's right.
All for a good purpose. And without science, we would not know any of that.
We would not.
Thank you so much, Fran, for telling us all about the macroinvertebrates and how wild they really are. And Rob, get on over here. And I'll just get Fran and Kimberly and Guy to get on over here as well. Have you had a good time?
Yeah, it's been fascinating. Really fascinating.
Learned some good science?
Yes, absolutely. I've learned about keratin and bottom breathing and
there is
there's
jelly
jelly
beans.
beans.
Jelly beans,
And
as always.
beans.
Jelly beans. And we need to get
Striped
Piper
marsh
up
frogs.
here
Oh,
as
Piper
well.
turtle,
Our
Piper
turtle.
turtle.
Yay, hooray. Piper, what was your favourite bit of today? Was it crawling through the net?
Probably, yeah.
I think you were
Was it the
enjoying it quite a
bottom
bit.
breathing bugs?
Can't get past the bottom breathing
Do
bug.
turtles breathe through their bottoms as well?
No, they have lungs.
They have lungs.
Okay.
have they?
They breathe at the
Yeah,
surface.
yeah.
There was some sort of oxygen intake through the cloaca at some point there. Anyway.
Yeah, no, you've been overreading.
Yeah.
Well, that's it for What a Wonderful World and looking at biodiversity. And these great scientists, let's give it to them again, from Griffith Uni, who are all studying and trying to work and make our place, our world a bit better and really make sure that our ecosystems are healthy, thriving, and if they're not, find out why so we can find out what we should be doing about it. And that's it from us on World Science Festival Brisbane today, pretty much. I do want to give a special shout out to all of you who are at home. Thank you so much. It's been so great having you here. I hope
Thanks,
you've been
guys.
yelling at the classroom if you're at school. And also, we had a special cohort of students from China. So, ni hao, ni hao. And I hope you had a really great time as well. And all the references to Brisbane weren't too much for you. Science and the World Science Festival has just been such a wonderful thing to have. As I said earlier, we are really sorry we can't be with you in person, but hopefully next year
Yes.
it'll all be back
Fingers
on.
crossed.
Absolutely.
Yeah,
In person.
and it's not too late to check in for some other great science shows we've got coming up on chemistry and physics in the next couple of days.
Yeah, definitely.
Yeah.
And keep studying science.
Yes.
Absolutely. There's
Any-
always something you can do to get involved. Yeah.
Any
There's-
final words on why science?
It's just that one day is not the same as the next. You know, there's always something new to look at. So there's new species to explore. We're still finding new species today. So there's always things to explore, new species to discover. So get out there and get involved. It's great.
From elephants to Stratty Island and
To bugs.
to bugs. Let's not forget the bottom breathing
And
bugs.
no, you can even have some caddises at home and make your own jewellery.
And the turtle rodeo. All right, thanks again.
Bye.
Great time. We'll see you again tomorrow maybe, and have a great rest of the World Science Festival Brisbane, and thanks to our education partner QPAC who made it all possible too. See ya. Happy
Bye.
science and